doing a test here at the end of the year, and I kind of want to wrap up, review some of these big concepts that we've been learning about. Going to share my the paper that you guys were working on, or it was posted on Teams. So scientific method, that's how we started off the year, and that you're going to find that's going to happen over and over again. Every year you're going to start off with a scientific method, and we talked about 7 steps, and I really hope you kind of learn how to use the scientific method, just to kind of help you with your day-to-day doing different things in your life. You might run into problems, learn how to fix things. It'll help kind of keep you disciplined a little bit when you're trying to solve a problem. But the first part of it was just observe. If you have a problem of some kind, and I always love to use the lawn test, or the lawn, trying to solve your problems in your lawn. But it can be anything. It could be you lost the bicycle on your bicycle chain. You observe that your chain is off your bicycle, and my questions is, how do I get it back on again? Your hypothesis could be, if I put the chain onto the first tooth and then rotate the pedals, the chain will ride back onto the sprocket of the bicycle. You do the test, see if that works. Your data would be, does it actually happen? Does the chain ride onto the sprocket? And your conclusion, it was successful, no, it was not successful. That would be an example of scientific method. So it would go observation, questions, hypothesis, test, data, conclusion, and if you were doing a real scientific research, you would publish your results. But most likely, if your yard is looking in bad shape or something like that, and you decide that you observe your yard is dying one part, and you ask the question, why is my yard dying in one section? And you come up with the hypothesis that the yard needs fertilizer, and then you set up a test, you fertilize a section of the dead yard, and then your results after a couple weeks, your data would be that either the grass grew back, became healthy, or nothing happened, and you can make your conclusion. That was number 1, scientific method. And then the dependent variable in the yard and test. If your test itself is what the scientist would control, that would be your independent variable, and that would be the adding the fertilizer to the yard and comparing it to the undead section, just putting in a little bit in a test area basically of your yard, and the data would be whether does the grass grow. So that you don't know what the data is going to be, so that would be your dependent variable. The data would be, does the grass grow back, or does the grass get worse? That would be your results. That would be your dependent. You do not control the dependent variable. You don't know what's going to happen. So that's going to answer number 2 for you, is that it's independent variable is what the scientist controls. Dependent variable is going to be the results of your test. Number 3 is basically, I should have been a little clearer with that, but the test is the independent variable and the data is the dependent variable. So you're kind of repeating yourself there a little bit. Number 4, kind of get a little bit more straightforward. What's the difference between animal and plant cells? Animals, don't have a cell wall. Plants do have a cell wall. Plants have chloroplasts. Animals do not have chloroplasts. Plant cells do have mitochondria. And animals have mitochondria, but the animal cells do not have the chloroplast. So that's the difference between the two, is that one has chloroplast and one has a cell wall and the other one does not. Number 5, what was the difference between mitochondria and chloroplast? This is basically the same concepts as in the question number 4, except mitochondria is the energy, is where energy is created in the cell. It does the respiration. You're taking in oxygen into the mitochondria, and you are releasing the energy in the form of ATP. And the chloroplast is what creates this energy. It takes in the CO2 through the leaves of a plant, and it converts the CO2 into a sugar. And that's the difference between mitochondria. One is the actual energy, and the other one uses the energy. The mitochondria uses the energy. So that's the difference between the two. How do plants build themselves from the chloroplasts? Okay, this one's a little bit out there. Chloroplast produces sugar, and plants, the sugar, if you add bonds to that sugar, okay, it becomes cellulose or wood or fiber, and that fiber, again, can be turned into wood. And that's how a plant builds itself from its chloroplast. The chloroplast produces the sugar. You add the bonds to the sugar. Okay, the bond that becomes cellulose. It becomes the structure to the plant. Okay, so that's how things are created through. That's how wood is created. What does the nucleus of the cell have in it that is important for running the cell? Okay, remember that there's codes, the genetic codes in the cell. It's called DNA. The DNA has the information to tell the ribosomes in the cell what type of protein to make, and the proteins get combined in the Golgi apparatus, and that then forms the tissues, or forms the components of whatever is being made in the body. What are proteins constructed? Where are proteins constructed in the cell, and what is the monomer that is used to make them? Okay, proteins, again, are made in the ribosome, and the monomer that's used to make them is amino acids. What organelle processed proteins and shipped them out? Okay, that after the proteins are made, they get shipped to the Golgi apparatus. And from the Golgi apparatus, they're processed, combined, and they are shipped off to whatever they're being used for, and being whatever's they're making, contributing to. What were the 3 molecules needed to construct DNA? The deoxyribose was the sugar. And remember, you have like a DNA is kind of on a dual skeleton structure of the deoxyribose and a phosphate group, and the DNA information is stored in the bases between these 2 structures, these helixes that are made from the deoxyribose and the phosphate. But the bases are, and I'm not going to make you guys know guanine, thymine, adenine, cytosine, but those bases in different, organized in different ways, in different patterns, is what codes for the information that gets sent off to the cell, or excuse me, the ribosome to, as in the form of mRNA, to be turned into protein. So those are the first 10 questions of our review. And I'm hoping you guys can remember some of the stuff that you've learned already. And so it's not too confusing, and hopefully that helps.